I'm Frank Furnari, and it's my pleasure today to introduce Dr. Tim Fenton, who's an associate professor of cancer biology in the School of Cancer Sciences at the University of Southampton in the UK. I've known Tim for about 18 years now. He was a postdoc in my lab 18 years ago, where he studied the mechanisms of resistance to therapeutics in glioblastoma, in particular, therapeutics that target the epidermal growth factor receptor. And it was about this time that there were a number of clinical trials going on where EGF receptor inhibitors, first and second generations, were being tested, but they had been failing miserably in clinical trial. And Tim decided to try to figure out what the cause of this was. And one of the mechanisms that he discovered is that the PTEN tumor suppressor gene was being modified on a particular tyrosine, tyrosine 240, and this was being facilitated by fibroblast growth factor receptor. And so this outstanding discovery is actually a large part of my lab right now. We're actually going forward looking at the mechanism behind this, but what it turns out to be is that when PTEN is phosphorylated on tyrosine 240, it actually binds to chromatin and plays a significant role in repair of double-strand breaks. And that's part of the mechanism why the tumors become resistant to drug as well as standard of care therapy. So in 2011, Tim left my lab for his first academic appointment back in the UK at the University College of London, where he switched gears and started working on head and neck and cervical cancers, in particular how HPV played a role in the genesis of these particular type of malignancies. In 2017, he moved to the School of Biosciences at the University of Kent. And as of 2019, as I mentioned earlier, he is now an associate professor at the University of Southampton, where he has been pursuing how cytosine deaminases, the APOBEC enzyme family, which he'll tell us about today, actually play a role not only in mediating resistance to retroviral infections, but also are causative in terms of tumor progression because they're mutagenic, I'm sorry. And with that, let me welcome Tim to our university, to the Stanford Stem Cell Consortium, as he presents his topic of APOBEC3 enzymes from retroviral restriction factors to cancer drivers and beyond. So welcome, Tim.
Thank you very much, Frank, for that very kind introduction and for the invitation to come and share some of our more recent work with you today. It's an absolute pleasure being back here in San Diego in January. It's significantly warmer than Southampton at the moment. And my main challenge, I think, is going to be to try to keep you all awake in those very comfy-looking seats that you all have. So, yes, I shall try and gain your full attention here. I'm going to talk about our interest in the APOBEC3 enzymes. I find these to be really fascinating enzymes. So a little bit about the background there, and I'm going to share some unpublished work with you today that I'm interested in getting your thoughts on too, certainly. So just one disclosure, and we will go into the talk. So as I say, first, something about the known roles of APOBEC3 genes, why we're interested in them, how we came to be interested in them, and then a story from the lab that is certainly an ongoing story. So here we go. This is just a diagram of the main thing that APOBECs do. They catalyze the deamination of cytosine bases in either single-stranded DNA or in RNA, producing a uracil base. And humans have 11 of these enzymes. And in particular, my talk today will be focusing on the 7 APOBEC3 genes, which lie in tandem on chromosome 22. And they've evolved very recently, really. So in humans, we have 7, for example. Mice only have 1 APOBEC3 gene. And my talk really will be focusing on APOBEC3A and APOBEC3B, neighboring genes very similar to each other, at least to outward appearances. So this is the kind of canonical or prototypical function for APOBEC3 enzymes, which was shown by Mike Malim and others pretty much 20 years ago now. APOBEC3G in particular gets incorporated into newly synthesized HIV virions, and it gets packaged together with the viral genome. And then when a new cell is infected, a new T cell, then and you get reverse transcription of the HIV RNA genome, the APOBEC3G essentially attacks that nascent cDNA, which is single-stranded, of course. And the effect that has is hyper-editing of the HIV genome, which renders it incapable of replicating. Of course, HIV continues to infect people, and the reason for that is that it has taken another step in the viral arms race, and lentiviruses have evolved a protein called VIF. And what VIF does is it targets APOBEC3G for destruction by the proteasome. So if you remove VIF from HIV, it can't replicate in human cells because the HIV is hyper-edited by APOBEC3G. So a very important antiviral role for APOBECs. And the different members of the 7-gene APOBEC3 family in humans are able to target a wide range of viruses. So some of you may have seen a recent literature on APOBEC3A, potentially, possibly APOBEC1, hyper-editing SARS-CoV-2 genomes, so that you can see a distinctive pattern of mutations that one can tentatively ascribe to the action of APOBEC on the genome. So why are we interested? So as Frank mentioned, I'm a cancer biologist. Several years ago now, this is really the discoveries that were made in 2013, and many of you will have certainly spent time looking at these, particularly those of you who've gone to Ludmila Alexandrov's talks, so I won't labour the point, but the realisation really in 2013 that one or more of the APOBEC3 enzymes are responsible for generating a large number of somatic mutations in a wide range of human cancers. And they produce a very distinctive pattern of mutations. The APOBEC3 enzymes have a preference for deaminating cytosine with a T in the immediate 5' position. And what happens if you have replication over that site before basic scission repair can be enacted and the C can be replaced with another C, you get actually C to T transitions or C to G transversions, and it appears to depend on the nature of the polymerase that's doing the replication across that site. In some tumours you see more C to T, and some tumour types you see more C to G. But both of these mutation signatures can be ascribed to the activity of one or more of the APOBEC3 enzymes. So these were some of the key papers that really showed that back in 2013 from Ruben Harris's lab, who was at Minnesota, now at UT San Antonio, and Dmitry Gordenin over at the NIEHS over in North Carolina, and also Mike Neuberger's group and Mike Stratton in Cambridge. So APOBECs are ostensibly there to counteract viral infection, but it would appear that in cancer there is this off-target activity. And for various reasons, this can really be ascribed to the activity of APOBEC3A and or APOBEC3B, and it appears that in different cancers both can play a role, with APOBEC3A being the primary cause of these mutations, but nonetheless APOBEC3B certainly causing mutations too. So that kind of summarizes some of what we know about these enzymes and how they function. So also, again, just showing the mutagenic effect of that deamination of cytosine if basic scission repair is not able to act before the site is replicated over. Again, if those of you who've been to Ludmil's talks will be used to seeing This figure, I expect, or versions of it, this just shows these two mutational signatures, 2 and 13, which I've put red arrows against, and the tumour types in which they're seen. And apologies, this is very small, but the main point I want to make here is that you particularly see enrichment for APOBEC-induced mutations in epithelial cancers. So squamous cell carcinoma is a particularly enrichment for these type of mutations. And this is where we came in, really. Around the time when these papers were coming out, we had been looking at genes that are overexpressed in HPV-induced cancers versus other cancers to try and see if we could pull out a set of genes that were important for HPV-mediated tumorigenesis. And this was work being done by Anka Chakravarti, who was a fantastic Ph.D. student with me and is now a postdoc in Toronto. He actually derived a gene signature for HPV-associated cancer. And what we were able to do, particularly looking in head and neck cancer, where you have two distinct etiologies, you either have HPV causing the cancer or you have a sort of smoking and drinking associated etiology. The smoking and drinking one is on the way down, but the HPV associated one is on the way up, and it's been described as a cancer epidemic. We can make a good comparison between the two types of cancer, and this is just a graphical abstract taken from a paper that we published back in 2014, where we showed that there's a strong enrichment for APOBEC3 mediated mutations in the HPV positive subset of head and neck cancer. You also see it in HPV negative, but it's particularly strongly enriched in positive and in cervical cancer, which is all driven by HPV. And the key probably point we made in this paper was to demonstrate that another gene that I'd worked on with Frank, PIK3CA, so PI3 kinase, is actually mutated by APOBEC at two hotspots in the helical domain, and that is actually, so our work was the first to ascribe actually a driver role for APOBEC3 mutagenesis in cancer as so beyond purely generating passenger mutations, this links the APOBEC3 activity to the generation of oncogenic mutations. So that's where our interest really came in. The clinical significance of APOBEC3 deregulation in cancer is evident from studies looking at expression levels. So APOBEC3B, high expression of APOBEC3B is linked to poor prognosis in breast cancer. In lung cancer, tumours with high levels of APOBEC-generated mutations tend to do worse. This is TCGA data on the left here. But on the right, you can see something that's becoming apparent in the era of checkpoint blockade and immunotherapy, that tumours with more mutations tend to respond better. They've got more neoantigens there for T-cell responses, and so it flips round and you see a flipping of the survival curves. More APOBEC activity in non-small cell lung cancer, admittedly with a very small number of patients from this Cell paper, looks to be associated with a better outcome. Now, when we're thinking about the development of treatment resistance, which has been something that I've been very interested in since my time in San Diego, of course, there are different ways in which resistance can occur. And at the top here, this is showing the treatment selecting for the emergence of pre-existing resistant subclones within a tumour. But we know that, or the data suggests, the evidence suggests that in addition to the selection of pre-existing subclones when we treat cancers with, for example, targeted therapeutics, there can also be de novo mutagenesis occurring in what we call drug-tolerant persister cells. And in this process, APOBEC activity has been invoked as a way in which tumours can essentially gain more heterogeneity, more potential to evolve treatment resistance through acquiring de novo mutations. There are a number of efforts underway in academia and in industry to try and target APOBEC, so to inhibit APOBECs, with the idea that you might be able to administer an APOBEC inhibitor alongside a targeted therapy, for example, to try and suppress this emergence of resistance. Of course, that's predicated on the resistance coming from de novo mutagenesis rather than the selection of pre-existing clones, which you can't do much about there. And the evidence for this comes from some very nice work from Aaron Harter and colleagues at MGH, looking in the paradigm of targeted therapy for lung cancer patients who have activating mutations in their epidermal growth factor receptor gene. They get treated with EGFR inhibitors because these cancer cells are essentially addicted to the activity of EGFR. And Aaron showed very nicely in vitro systems that you can get both mechanisms at play here. The emergence of resistance in drug-tolerant persister cells, in addition to the selection of very rare pre-existing resistant subclones. And this is from a preprint from their lab, just showing that this is in the context actually of a different targeted therapy which targets a particular fusion protein in lung cancer, just showing that in a treatment-naive tumour there was very little appearance of APOBEC-induced mutations, but in the many treatment-resistant cancers that was subsequently sequenced when the patient developed resistance to to the crizotinib in the first instance and then to other ALK inhibitors later on, you could see a very strong enrichment for APOBEC signature mutations in these tumours. So really looking like APOBEC's playing an active role in that development of resistance. And there's some nice work in cell lines to complement that in that study, including the demonstration that APOBEC3A is actually induced by EGFR TKIs. One interesting challenge in the field is that we see very little correlation between the expression levels of APOBEC enzymes and the mutation signature. So it's kind of, there's a lot of currently hand-waving about episodes of APOBEC expression and mutagenesis, but by the time the tumour is actually resected, often the levels are very low. But it kind of makes sense. A tumour is likely to lose fitness, the tumour cell is likely to lose fitness if they have a high level of this very mutagenic enzyme on constitutively. So the thinking is that there's episodes of APOBEC-induced mutagenesis. Just before I get on to our recent work, I wanted to show you evidence that there's function for APOBEC beyond this hyperediting of viral genomes. There appear to have been additional functions that have evolved for APOBEC3A and APOBEC3B. This is the graphical abstract from a very interesting paper published by a collaborator of ours, Simak Ali, at Imperial College London, several years ago now. But there's been some very nice follow-up on it very recently, preprint, showing, this really surprised me when I saw this, be interested for your thoughts on this, but showing that APOBEC3B associates with the estrogen receptor, goes to estrogen receptor target genes, and actually the deaminase activity of APOBEC3B is required to make double-strand breaks in the promoter that recruits the DNA repair machinery, and that has the role of remodeling chromatin in these promoters. Now, it seems like quite a wacky way for a cell to enact induction of gene expression to make double-strand breaks in this way, but the data are very solid, and as I say, there's been follow-up study recently showing this is probably a wider-ranging phenomenon than just with estrogen receptor. So that's one way in which APOBEC3B appears to have acquired a role in transcription, and that may actually explain its role in ER-positive breast cancer, so that poorer prognosis associated with higher APOBEC3B expression may actually be related to this function as opposed to its mutagenic function. With APOBEC3A, there's recent work from a group at Yale showing that APOBEC3A in a deaminase-independent fashion can go and sit on the interferon-stimulated gene promoters on the interferon response elements and actually repress the expression of ISGs. So APOBEC3A itself is an interferon-stimulated gene, but it appears to enact a kind of negative feedback loop to dampen down that interferon response by acting as a transcriptional repressor. So both APOBEC3A and APOBEC3B, these culprits for somatic mutagenesis in cancer, appear to be playing important roles in not only antiviral responses, but in this case, regulation of gene expression. So, as promised, I'm going to share some of our more recent work with you today. Now, we are very interested in understanding how APOBEC3A in particular, but also APOBEC3B, as I'll show you, is normally regulated and what it may be functioning, or what it may be doing in keratinocytes. And the reason for this, as I mentioned earlier, a lot of the tumours with the highest APOBEC-induced mutation loads are squamous cell carcinomas arising from squamous epithelia. So we want to look, rather than trying to look in cancer cells, we're looking in normal keratinocytes, how things should be regulated, with the aim of understanding what's going wrong during tumour development. Now, APOBEC, and the work I'm going to present is being carried out by two excellent postdocs in the lab. Nikki, who did her Ph.D. with me, is now a postdoc on the wet lab side, and Ian, who is an excellent bioinformatician. So, APOBEC3A is actually celebrating something of a birthday this year. It's 30 years ago that the group, Rasmussen and Sellis, published this paper showing an inauspicious looking spot on a 2D gel. Some of you probably have not seen 2D gels before. Isoelectric focusing in one dimension to separate proteins by their isoelectric point, and then SDS page and to separate them by size. And what this group observed was a protein, or a group of proteins, that were overexpressed in keratinocytes taken from psoriasis lesions. And they showed those same proteins, they could induce them if they treated keratinocytes with forbolester, so PMA, which is also well known as a tumour-promoting agent. And Spot 2116, they named formalin-1 because it came on with forbolester treatment. And several years later, once the APOBEC3A gene had been cloned, another problem we don't really wrestle with very much these days, it was appreciated that formalin-1 is actually APOBEC3A. So APOBEC3A, highly overexpressed in psoriasis and potently interferon, well, it is interferon inducible, but potently forbolester inducible in keratinocytes. Okay. So to kind of summarize where we're at the moment, APOBEC3A, for reasons that are probably too niche to go into at this point, is likely the primary driver of somatic mutations in cancer, but its expression is often very low to undetectable in the tumour biopsies. So there's a weak correlation between expression and mutation signature load. And also, looking in bulk tumour data, where a tumour is obviously a group of many different cell types, can be quite misleading when looking at APOBECs. There's a lot of APOBEC3A expression in macrophages. There's a lot of APOBEC3G expression in T lymphocytes, for example. So when one's looking at RNA sequencing data from bulk tumours, it's very difficult to know what's actually expressed in the tumour cells and what's rather expressed in, for example, infiltrating immune cells or stromal fibroblasts, for example. So we've taken advantage of a lot of single-cell RNA sequencing work going on in our department at the moment. This is with our close collaborators, Professor Gareth Thomas's lab and Emma King, who's a head and neck surgeon. And what I'm showing you now are single-cell RNA-seq data, and this is from normal oral mucosa. So these are matched normal samples taken at the time a resection was done for a patient with head and neck cancer, in this case tonsillar cancer. And they take normal tissue from the contralateral tonsil. This is just the tumour cells. So Gareth's really interested in fibroblasts. So he said, we've got all this single cell data. I'm just interested in the fibroblasts. You go and have a look at the tumour cells, which in this case, normal epithelial cells, which was very gracious of him. And what we saw for various reasons, having looked at a number of other cell states, including cell cycle, for example, we got interested in the differentiation status of these cells in this single cell data. And what we observed was that APOBEC3A expression was largely confined to a group of differentiating cells in these normal normal samples from the oral mucosa. The same is true when it's important with single cell sequencing as well to look at what's happening on an individual case-by-case basis, just so that you're not seeing an effect that might be skewed by a few cases showing a pattern. And we can see that in individual cases, again, the APOBEC3A expression is correlated with differentiation genes rather than with genes expressed in the basal keratinocytes, things like P63. And that's true both in normal samples and in tumour samples. And I should say in the tumour samples, this was done very carefully, so the surgeon selected regions right from the core of the tumour, so we're not looking in the tumour cases at normal epithelium at all, we're looking at just tumour cells there. So we were very interested in looking at APOBEC3A and B because there's been a lot of controversy and kind of debate out in the field over the years about which is really the key mutator. And this is obviously important for people thinking about trying to develop inhibitors. Which one do you go after? Do you go after one or the other or both? And often from bulk data, you kind of see just a kind of general, they appear to be somewhat correlated, and that's all we can really say. So we were very interested to look at what this looks like on a cell-by-cell basis, and what we saw was something that I think, at least, is very interesting. You see the occasional cells where they're both there, but actually the expression patterns are distinct, and that's probably clearer if I show you the pathway analysis of the genes that are most highly correlated on a cell-by-cell basis with APOBEC3A or B. With APOBEC3A, what we see, apologies if this is rather small, but you'll have to believe me that I'm reading to you correctly, that it's genes associated with keratinocyte and differentiation. Okay, the differentiation process in these stratified epithelia. So that's the genes that are co-expressed with APOBEC3A, and that shouldn't be surprising to you because it's kind of what I've just already described to you. But now we go to APOBEC3B and we see something very different. Here we see genes that are essentially involved in the cells transiting from G2 phase of the cell cycle through to mitosis. This is very interesting to us because, as I'll show later, we were quite easily able to get rid of APOBEC3A. We were able to use CRISPR to make APOBEC3A knockout keratinocytes, no problem. Applying the same techniques, strategy to APOBEC3B, we've been unable to recover APOBEC3B knockout keratinocyte clones, and we do think that APOBEC3B is playing a key role actually in cell cycle progression in these cells. In breast cancer, Simak Ali observed if he reduced APOBEC3B expression with an shRNA, he also see a decrease in proliferation. So we're looking into the mechanism by which that's happening. The same is true in the tumour cells as well. So I've just shown you data from the normal mucosal samples. This is from the tumour samples. The same thing. We see APOBEC3A in the more differentiated tumour cells and APOBEC3B co-expressed with genes involved in the transition to mitosis. If we look at a panel of HPV positive oropharyngeal squamous cell carcinomas, so that's essentially tonsil and tongue-based tumours, that is really the site at which HPV has a strong involvement in the head and neck. You can see again, this emphasises the point that the expression pattern of APOBEC3A is closely related to the expression of genes that are expressed in differentiated cells, whereas APOBEC3B, actually the gene that is most highly correlated with is Ki67. So very much, classic marker of cell proliferation. This is also true in esophageal squamous cell carcinoma from a large data set that we analysed. I should point out that aside from data on the esophagus and the head and neck, we looked at quite a number of other single-cell RNA-seq data sets, for example, from lung cancer, breast cancer, and others, and there's just not enough APOBEC3A expression in those data sets to see it. So we know that head and neck and esophagus have a lot of these APOBEC signature mutations, so maybe it makes sense that we can see more of the APOBEC3A in these particular tumours. So these are the ones that we've been able to look at and see this relationship so far. Now, we then wanted to move, obviously, to an experimental system, and we work with a cell line called NICs, which were given to us originally by Professor John Dorbar in Cambridge, who is an expert on HPV. NICs was originally isolated about 20 years ago or so from neonatal foreskin, so skin keratinocytes, and is spontaneously immortalized. But they have a diploid genome, so it's nice for doing CRISPR. You're not worried about trying to knock out 6 copies of your gene, as can happen in cancer cell lines. And it importantly retains the ability to differentiate in culture. So you can make rafts and it will reform a stratified epithelium, a keratinizing epithelium, and we can grow them to high density and induce differentiation that way as well. In fact, HPV can complete its life cycle in nicks, and that's completely dependent on differentiation. This data shows that what John's lab were able to do was they did single-cell RNA sequencing of nicks grown to high density. What happens when you grow nicks to high density, even in a standard 2D culture, is you start to get a second layer of nicks growing on top of the first layer, and those cells are more differentiated. The bottom layer are basal-like. So we have the different cell types arranged here, where we have the basal nicks up in the right. We have these cells here, which are in S phase of the cell cycle, in blue. These cells here are in G2M. And then as we go down this continuum here, we're getting more and more differentiated. And what we can see, which is very useful for us, is that in our cell model in the lab, we see the same thing as we saw in the tissue samples from human patients. So the APOBEC3A is all down here in the more differentiated cells, and the APOBEC3B is largely in the G2M phase of the cells that are cycling, and a little bit here in S phase. So that was useful for us because we thought we had a reasonably relevant model system to be looking at. Now, going back into the lab here, what we were able to see, or Nikki was able to do, was develop some reasonably simple assays in which we could see at least the onset of differentiation in our NICS cultures. So differentiation-related genes coming on. In this case, I'm showing you involucrin and cytokeratin 10. So if we starve our cells, Nikki starves them for 40 hours, we see an induction of involucrin and cytokeratin 10 expression. We also see it, as I say, if we grow the cells to high density in the middle there, and if we add an EGFR inhibitor. So I've stayed true to EGFR in some respects at least. And this is interesting because it's similar to what Aaron Harter has observed in lung cancer cells. Treat them with an EGFR inhibitor and you induce APOBEC. In this case, we're inducing a differentiation response here by treating the keratinocytes with an EGFR inhibitor, which is something that's been long established. It's well known that if you inhibit EGFR, you'll induce differentiation. So these three conditions, we see the induction of the differentiation genes, and sure enough, we see induction of APOBEC3A expression under these conditions. So this all ties in nicely with what we're seeing in the clinical samples, in the single cell data from NICS, and now we're able to manipulate the expression of APOBEC3A under these conditions. So far I've shown you that APOBEC3A is expressed in differentiating keratinocytes, while B is expressed in proliferating cells, so they're in different cell states. Next, we wondered what the function of APOBEC3A is in this context. Why is it being turned on during differentiation? And this is really kind of where we're at the moment. We've conducted a lot of RNA sequencing, and we've seen, actually, rather interestingly, that what happens when we knock APOBEC3A out is we have an apparent loss or a defect in differentiation. So again, this list of processes here from the gene set enrichment analysis shows that what we're losing when we knock out APOBEC3A is keratinocyte differentiation. Of course, with CRISPR, one has to be mindful of have we got odd clones here that are doing different things. We did the RNA-seq from 2 independent clones of wild type and A3A knockout, and we've got additional clones where we see very similar. This is just showing the growth curve. So what we immediately observe, and it's obvious when working with the cells in the lab, is that the knockout cells grow to a higher density before they stop and start to differentiate. So you can see here the slowdown in growth happens at a higher cell density with the knockout cells, and we see the induction of involucrin and cytokeratin 10 expression at a higher cell density in the case of the APOBEC3A knockout. Quite dramatically, if we add EGFR inhibitor to the knockout cells, they don't show the same response with the involucrin and cytokeratin 10 induction. And again, if we starve the cells, so another way in which we induce differentiation, we really blunt that induction of involucrin and cytokeratin 10. And I'm always paranoid that things are artifactual until we've shown it a few different ways. So we've really struggled to find an siRNA sequence that hits APOBEC3A but not B. There's lots that are advertised to do so, and our usual kind of workhorse with siRNA is the on-target plus siRNAs from Thermo, the smart pools. Well, this smart pool isn't so smart. So in that case, the on-target plus smart pool hits both A and B very strongly, and I think there's quite a lot of potentially red herrings out there from people looking at APOBEC3A and B with shRNAs and siRNAs, but I'm going to leave it there and not name any names. But this is a sequence that hits A and not B. You have to believe me on that because I've not shown the B data. But what we see when we hit the cells with A3A siRNA is that we again see a reduction in the cytokeratin 10 induction, shown here, in this case when we're adding EGFR inhibitors. So we do think this is a real phenomenon that getting rid of APOBEC3A specifically gives us a differentiation defect. The other thing I wanted to show in terms of what we see at the level of gene expression in these cells, and maybe this is related, and I'm no expert on skin differentiation. I'm trying to become one, but if any one of you has thoughts on this, I'd be most interested. I mentioned the PNAS paper earlier, where they show a role for APOBEC3A in repressing ISG expression. And this shows a group of the genes that they identified being most differentially expressed in wild type versus APOBEC3A knockout. In this case, they used T cells and hit them with interferon. And this shows a group of ISGs whose expression is increased in the knockout cells. We see the same thing in our keratinocytes. So if we either look at if we hit our keratinocytes again with the EGFR inhibitor, and we see very similar when we starve the cells, we get an induction of these ISGs. So I'm showing 3 of them here that we've looked at by QRT-PCR. And you can see that if we knock down A3A, again with our A3A specific siRNA, we get a large increase in the production of these ISGs. So our data are certainly consistent with those of the Yale group. And so we are currently working on the working hypothesis that this regulation of transcription by APOBEC3A may extend beyond these ISGs, in the keratinocytes at least, and we're looking at some of these differentiation genes to see whether it may directly be modulating expression of those. Now, that's all in the case of differentiating cells. What Nikki was very interested to do too was to see what the inducibility of the APOBEC3A gene looks like in starved cells versus asynchronous growing cells. And this was really interesting. So shown here, the scale's very big for reasons that will become apparent shortly. This is what happens. This is the increase in APOBEC3A expression that Nikki sees if she adds forbalester, a known inducer of APOBEC3A, and interferon, another known inducer of APOBEC3A. She gets a nice increase in expression from adding to asynchronous cells. But she then, just out of interest, and probably because we were interested in EGFR as well, added growth factors back to the starved cells to bring them back into the cell cycle. And then she saw this enormous induction of APOBEC3A. This is way beyond anything we've seen before. And it actually gets up to levels that you can see if you start to play around with transfecting A3A into cells. And so this we see nicely an increase at the protein level, shown here, Western blotting. It's unusual to see, we'd not seen conditions under which we could really detect the APOBEC3A expression with native antibodies. We'd had to use CRISPR to put an HA tag on the endogenous gene to be able to follow it. But now we can see it very nicely with the antibody against the endogenous protein. This just shows the level of induction we get when we starve the cells, which I was showing you earlier. But look what happens now if Nikki adds PMA and interferon to the starved cells. We have a much stronger induction of APOBEC3A when she adds PMA and interferon to starved cells than when she adds it to the asynchronous growing cells. And then we can even increase that further by combining growth factor stimulation with PMA and interferon. So what we think is that this initial increase in APOBEC3A expression that we see upon various ways of inducing a differentiation, starvation, EGFR inhibitor, high confluence, is not only increasing APOBEC3A gene expression, but it's making it very potently inducible by other factors. We can see that if we need EGF to really give the full response. These cells grow in a mixture of serum, EGF, insulin. They're quite picky, a number of other things, and we grow them on mouse fibroblast feeder cells. And we can block this with an EGFR inhibitor. So we've got these two things going on now. In the first instance, in proliferating NICs, if we add an EGFR inhibitor, induced differentiation, we increase APOBEC3A levels. But in the case of once we've starved cells and we re-add growth factors, we have an EGFR dependent spike in A3A expression, which is up to kind of what we're thinking of, we're calling these kind of pathological levels, the levels that one would see in, for example, psoriasis lesions, and potentially spikes that would occur in cancer, during cancer development that might be giving these somatic mutations. If this is not some funny quirk of NICs, we see the same thing if we use primary keratinocytes, very similar induction of expression, and again inhibited by an EGFR inhibitor. And again with the primary cells, the opposite's true when we starve the cells. If we inhibit EGFR, we lose the initial increase in A3A that we know is important to give this subsequent big burst in expression. So I know it's a little bit hard to wrap one's head around. We've got EGFR acting in the opposite fashion in the two different contexts. But that's what we see. And we see in both cases, this is proceeding via the RAS-MAP kinase signaling arm of the pathway. So it's, if we use a MEK inhibitor or an ERK inhibitor, we see exactly the same as if we use an EGFR inhibitor. We see nothing if we use an AKT or a PI3 kinase inhibitor. MCF10A, so different type of epithelial cell. Now this is mammary luminal epithelium. We see the same thing, but it's at much, much lesser magnitude. So there's something we think specific going on in keratinocytes. We've tried a number of different cell types, and keratinocytes really are the ones that give us this very strong phenomenon. And we don't see it with the other APOBEC3 genes. So this is a specific effect among the 7 APOBEC3 genes to APOBEC3A. This is, as I say, this paradigm in which we are starving the cells, then releasing them back into the cell cycle, and in this EGFR-dependent increase in A3A expression. You can see the protein level here. This is a deaminase assay that we do on cell extracts using fluorescently labeled single-stranded oligonucleotide. When APOBEC deaminates its target cytosine in this oligo, we then use sodium hydroxide, heat the oligo, we get cleavage of the oligo because of the presence of uracil DNA glycosylase, which has removed the uracil to create an abasic site, which is then cleaved. We then see this lower molecular weight product. This is the oligo that's been cleaved, so it shows the deaminase activity. And we also see, using a digital PCR assay developed by our collaborator Remy Buisson, who's just up the road at UCI, we see a very strong RNA editing activity too in these cells. So this is looking at a particular mRNA called DDOST that Remy showed was edited in tumours with high APOBEC3A expression. We see a very strong RNA editing activity. So we've taken samples from the wild type and knockout cells. We've conducted some more RNA sequencing, deep sequencing and exome sequencing, which we're using now to identify globally the RNA editing events that are catalyzed by APOBEC3A, because it's possible we see strong RNA editing. We don't see genotoxic effects under these conditions as measured by gamma H2AX, which you do see if you transfect A3A into cells. And so we're asking the question whether actually the RNA editing activity here of A3A may be important in mediating these effects that we see. As I say, in the PNAS paper, they showed it was a deaminase independent effect. The other thing that we are doing is adding back the wild type or deaminase dead mutant of APOBEC3A into the knockout cells to see if we can tease out whether this is deaminase dependent or independent effect, and that is ongoing. So I wanted to share this kind of model with you. This is just something that I'm thinking about at the moment, and would be kind of certainly grateful for thoughts on. I'm wondering whether we see, so I should say that APOBEC3A is about 10 times more potent as a deaminase than APOBEC3B. We know the APOBEC3B is on encycling cells, at least in G2. A3A, as far as we can see from the single-cell RNA-seq data and our experiments in NICs, is not being expressed in cycling cells. I haven't shown you today, but if we use a CDK4/6 inhibitor to block the cells in which we get the big induction of A3A from going into S phase, we still see that strong induction. So that's happening in G1, I think, in a set of cells that are not then going on to replicate. The cancer mutagenesis catalyzed by A3A is happening at the replication fork during DNA replication on the lagging strand, where you get exposure of single-stranded DNA. So there's a lot of work has shown that. So a cell would not really want to have A3A on as it goes through S phase. So I wonder whether this is a kind of low-risk tactic by the cells, that they don't risk expressing A3A in the basal cells, which have proliferative potential. But once the cells have initiated terminal differentiation, you're not worried about accumulating mutations, because the cells are going nowhere. They're just going to die. They're on that pathway of terminal differentiation. So maybe there you can afford to have more APOBEC3A and a more potent antiviral response, because you're not worried about accruing mutations. This is just an idea, and we certainly need to see whether we can to prove that or disprove it. But that's a kind of way in which we're thinking about this. But the other side of this is that if you have an injury, a wound healing response, which actually, if you look at the genes that are upregulated when you re-add growth factors to starved keratinocytes, you see a lot of wound healing genes coming on, then this may be where you have problems. And this might be some of what we're seeing in cancer, is when you then have A3A high cells potentially proliferating. So I think it's wrong place at the wrong time for APOBEC3A. It shouldn't normally be on in cells that are proliferating, but if it is, then that's where we could have the risk of mutagenesis. So normally should be restricted to the differentiating cells, but if it's on in cells that are cycling, then that's where we may have the problems. And I'm reminded of many people have described this, but this is taken from a particular review by Harold Dvorak, New England Journal, tumours being described as wounds that do not heal. So these processes of a wound healing response going on. Again, that's speculation, but that food for thought maybe, that this could be the conditions under which we see the problems with APOBEC3A. So I've shown you that APOBEC3A is expressed in differentiating keratinocytes. Hopefully that's been reasonably convincing, while APOBEC3B is expressed in proliferating keratinocytes. So this is all new. It's expressed, APOBEC3A, and strongly inducible in those starved cells that have exited the cell cycle. And we see the same thing in cells that we've made very confluent. APOBEC3A loss results in a differentiation defect, at least as measured by the expression of involucrin cytokeratin 10. We're not getting a full differentiation there, but those markers, and increased interferon-stimulated gene expression in the keratinocytes. So as I say, is this a mechanism by which cells are switching on APOBEC3A in conditions where they know they're not going to be undergoing DNA replication to get a stronger antiviral response? So that's what I wanted to tell you. Thank the people who've done the work. I already mentioned Nikki and Ian. Anka and Stephen led the work initially, where we showed the role for APOBECs in making PIK3CA mutations. Gareth and his lab for single-cell RNA-seq data from head and neck cancer. Remy for help with the digital PCR assay to look at editing. John and his group in Cambridge for the single-cell RNA sequencing data from NICs. And of course the patients who donated their tissue for this work, and the funders who funded the work. So thank you very much for your attention. I take any questions.
Great talk. Thank you so much. So two questions, two kind of, one on the line of the viral infection and the other one more on cancer. So stem cells are more resistant to viral infection, and especially in the skin, we know that the basal cells are much more kind of resistant to viral infection. And I wonder, based on your single-cell RNA sequencing, the APOBEC versus A, that there's really differential expression between those two, if you think there's anything to do with actually being giving the advantage of those basal cells to resist viral infection. The second question is more for the cancer. So as cancer progresses in basal cell carcinoma that you mentioned, as it progressed throughout development, then there is an EMT that happens to induce or to allow metastatic progression to happen. And I wonder if you see any kind of differential expression of APOBEC-B versus APOBEC-A in the basal cells versus the differentiated cells as well.
Great. So the first one, yes, I think, so B and A have been shown to be able to respond to different viruses, and it would be very interesting. So for example, HPV induces APOBEC3B expression through the actions of E6. But actually, the data that there are have implicated A as being a restriction factor for HPV. The data are not strong on that. So it's, I guess if B is doing something important in the basal cells to help with the restriction of viruses that may encounter there, and then for some reason there's a switch to A in the differentiating cells, it's a very interesting thing to think about. I mean, as you know, HPV goes into the basal cells, so I don't know. It's what we're just trying to get our heads around at the moment, and I think it would be great to have a discussion about that. The second question, so in terms of looking at EMT, we're just starting to play about with doing some siRNA against transcription factors involved in EMT, because we're wondering whether that's what we're seeing with the wound healing response. So it's coming up in some of the pathway analysis when we compare the starved versus the stimulated conditions, yeah. So that's a sort of beneficial side effect of what we've got from our RNA editing analysis for RNA-seq. So I think it could well be the case. We're looking at a gene called FRA1, so part of the AP1 complex that's involved in wound healing and DMT, because we see that being highly expressed in these situations, and it's downstream of EGFR2, so it's a good candidate, I think. Yeah, and we've done some 4C-seq as well, looking at additional cis-acting regulatory elements that may be switching on A3A to that strong degree.
Tim, have you done any conditional knockouts for these? I mean, I remember we had this conversation a while back, and you were trying to actually delete the whole locus. I mean, there's a number of APOBEC genes all in a row, and it's kind of complicated. Maybe you can comment on that.
Yeah, we'd like to. You have to be so careful with these genes because they're so similar to each other. For example, Affymetrix oligonucleotide arrays, I know people don't use them anymore, but they all the probes cross-reacted against the different APOBECs. There's a lot of antibodies that are not specific, and trying to design guide RNAs or SIs has been really challenging because the chances of hitting more than one are high. We haven't done conditional knockout. What we have,
but in the mouse, have you tried it?
Yeah, in the mouse, what we've done is we've replaced the single mouse APOBEC gene, which encodes a cytoplasmic enzyme. So mouse experimental tumors don't have this APOBEC mutational signature, and they're much more kind of mutationally bland than the human tumors. And so what we've done is we've replaced the single mouse gene with an entire fragment, about 210 kb, of chromosome 22 that contains the entire human APOBEC3 locus. So we have a humanized APOBEC3 mouse now, with them all under control of their own promoters. So we've got to the point of showing that the genes are expressed in a human-like fashion, and we're just doing some MLV experiments with a group at the CRIC, showing that we have actually functional APOBEC activity in splenocytes. And we've crossed that now, initiated crossing to a lung cancer model. So that we want to see if we're getting human-like tumors. And then on that background, we want to then knock out individual APOBECs and see what's happening. But yeah, that's, we're hoping
it's a long-term
it's a long
project.
and expensive project. Yes, but Genentech seem like they want to help us with that, so that might be a useful source of funds.
So philosophically, why do you think we have these genes? I mean, antiviral, or are there other things in evolution that they're necessary for?
The people who spend a lot more time thinking about the evolution of them than I do and have the expertise, the thinking is that actually the human, in higher primates than humans, the locus is expanded a lot to counteract endogenous retroviruses. So it seems like the ERVs are what have driven this diversification of the locus. Several of the APOBEC3 genes have been shown really important in controlling retroelement activity, and that's another thing we're looking at with the RNA-seq. That could be related to this ISG induction. I was having an interesting chat with Faye earlier over at the Cancer Centre about that. Yeah.
Well, there's no further questions. Just want to thank Tim once again for an outstanding talk.